So, hello, good morning. My name is Omar Bryant, and I'll be doing a presentation on the testing of COVID vaccines with animal models. So, a little background, a little outline of what we'll be discussing in this presentation. We have a background which will be about COVID in general, the different vaccines that are available, and the different strands of the COVID virus. Also, we'll be discussing an article by Corbett and all that is published in 2020, and that is like the COVID vaccine in mice. And we also dive into an article by Tien and his colleagues that was also published in 2020, which the topic of that paper is a SARS-CoV-2 spike glycoprotein vaccine candidate NVX-CoV2373. And then lastly, what we discussed will be takeaways from the two articles. So COVID in a nutshell. The new strand of COVID was first identified in Wuhan, China in December of 2019. And that strand was ended up transported over to the United States by February 2020 in Washington. They got its way to California and so on until it became a global pandemic that we are currently in. So there are three different strands of COVID. There's the SARS-CoV, the MERS-CoV, and then the SARS-CoV-2. The structure of the virus itself, it's COVID is non-segmented and has enveloped viruses that contain a single strand of RNA that ranges from 26 to 32 kb in length. The individual particles of the CoV-2, they're spherical shape with the diameter that's between 60 and 140 nanometers. Then there's also spikes that 9 to 12 nanometers that give it the appearance that COVID has the typical spherical spiky shape. As I was mentioning, it's spherical, and these are the spikes that appear. So COVID is commonly found in many species of animals. This includes camels, cattle, cats, and mainly and also bats, which the symptoms seen in animals are completely different from human. In humans, COVID commonly called it mild upper respiratory tract illness that ultimately could lead to death. Typically, when you have COVID, it's contagious for a 10-day period in which the symptoms that you may show vary from person to person. Some people could be have really bad shortness of breath and loss of taste and smell, compared to when I had it. I just had really bad fatigue. So the symptoms vary. So with the MERS-CoV strand, its full name is the Middle East Respiratory Syndrome. This strand is first reported in 2012 in Saudi Arabia, which that's why it gets its name Middle East, since Saudi Arabia is in the Middle East. That's where the name comes into play. And the common symptoms with this that goes along with this particular strand are severe respiratory illness, which including that is fever, cough, and the shortness of breath. And death could happen with this strand. The second strain that's common is the SARS-CoV, which SARS stands for Severe Acute Respiratory Syndrome. This particular strain was first reported in February of 2003 in Asia. In 2003, like this particular strain spread to over two dozen different countries, and some countries that are including this one found in the continent of North America, South America, and Europe. All right. And then lastly, the third strain is the SARS-CoV-2, which this is like the beta strain to the original SARS-CoV strain. And this one originated from bats. It's like you can see the structure of this strain, like it's the same as the normal strains. It's spherical and it has the spikes protein. Like that's what these like red things are. So yeah, there are currently 3 different vaccines, which are the Pfizer, Moderna, and Johnson & Johnson. Following the last dose, it takes 2 weeks for you to be fully vaccinated from COVID. So the Pfizer vaccine. This vaccine was made by Pfizer BioNTech company. This is a type of vaccine it's an mRNA vaccine. And with this vaccine, you need 2 shots. Each shot, the first shot is given 21 days prior to your second shot. And like the location in which they give you a shot is this area of your arm. So with the Moderna vaccine, the Moderna vaccine was made by Moderna Text Incorporation. The formal name for this vaccine is the mRNA 1273, which based off its name, it's concluded that it's an mRNA type. And with the Moderna vaccine, you get 2 total doses. But this time, with this vaccine, they're given 28 days apart rather than 21 with the Pfizer. But it's also given in the same area of your arm. So the Johnson & Johnson's Janssen vaccine was made by Janssen's pharmaceutical companies of Johnson & Johnson. The formal name for this particular vaccine is called the J&J-784366735. Unlike the previous 2 vaccines, this vaccine is actually a viral vector vaccine. With this vaccine, you only need 1 dose, and it's given in the same location as the previous one. After this singular dose, 2 weeks later, you're fully vaccinated. But with this vaccine, recently there was recalls because this vaccine caused really bad symptoms in a really small portion. So it wasn't until like April, until like recently, like a few weeks ago, people are allowed to receive the Johnson & Johnson vaccine again. All right, this leads us into the first article that will be discussed in this presentation, and this is the title of the article is essentially COVID vaccine in mice, and this was done by Cobat et al. Or Cobat and his colleagues. So essentially, like for this experiment that was ran, there's 4 different groups of mice. So like one group was like the control group, and they received a phosphate buffered saline rather than the actual COVID vaccine. And then the next 3 groups received different size doses of the COVID vaccine. The size doses that were given were 1/100th of a microgram, 1/10th of a microgram, and 1 full microgram. Like keep in mind, like with a microgram, it's significantly smaller than like a gram or like a milligram or a kilogram and all that. It's like microscopic size doses. So the mice that were involved in this experiment, they were assessed for like their body weight, how much lung hemorrhage they showed, the neutralizing antibodies that were found inside their body, and the lung viral load. So here's like the first figure, like figure 1 shows like the MERCoV2P mRNA protecting mice from lethal challenge. So the mice were given a vaccine at 0 and 3 weeks. So if you look at panel A, like this shows like after 4 weeks, like the number of neutralizing antibodies were measured in each group of mice. And the mice that received the 1 microgram dose of the vaccine had the most neutralizing antibodies. So if you look at the way the body weight was measured, it measured off the percentage of the body weight of like the mice at the beginning of like the first dose of the vaccine. So the mice that had a control that received like the control group, like the PPS, had within like the 8-day span had the biggest loss in percentage of body weight, but started around 100%. And after 8 days, the mice were weighed in at 70% of their original body weight. So with the lung viral load that is measured, the mice that received the control had the most PEU per lobe out of the different groups. If you look at the mice's lungs are measured for like hemorrhaging, the group of mice that received the control had the most amount of hemorrhaging. So in this figure, it shows like the mRNA 1273 vaccine protecting mice from upper and lower airway infections from the SARS-CoV-2 strain of COVID. So what we have in this one, they're still like the different groups of mice. Like there's a group that's given 2 doses of the control, which was the PBS solution. Then the mice that's given 2 doses of, also given 2 doses of the mRNA 1273 vaccine, and there's a group of mice that are given just a single dose of the mRNA 1273 vaccine. So one of the groups of mice that are given 2 doses of the vaccine, like the group that was given the control, not was shown to have a great amount of PEU per lobe compared to mice that are given 2 grams of 2 doses of various size vaccines. The mice that also given the 2 doses in control also had the most PEU per turbinate, which turbinate's like located in the nasal cavity of the mice. And then the mice that are given just 1 dose, the mice that was given a tenth of a dose, a tenth of a microgram dose, had the most PEU per lobe, which is different from when they're given 2 doses. The 2 doses of the control had greater, but when it's just 1 dose, the mice that would give a tenth of a gram had the most. Finally, like the figure 4D shows the amount of hemorrhaging in the lung sections of the mice at day 2 and then day 4 after like receiving the vaccine. So we have the control. This is from the 100th of a microgram. This is from a tenth of a microgram, and this is from a whole microgram of the vaccine. As you see the group of mice that received the PBS, which is the control, had the most hemorrhaging after days 2 and after day 4. As this goes into our next article that we discussed, the next article is the SARS-CoV-2 spike glycoprotein vaccine candidate NVX-CoV2373, immunogenicity in baboons and the protection in mice. And this was done by Jing Hien Tian and his colleagues. So the purpose of this experiment was to create a COVID vaccine that provides herd protection from COVID, protection from the virus, which could stop this pandemic that we're currently in. So the design for this experiment was first, viral E6 cells were maintained in the minimum ego medium, which is known as the MEM. A 10% fetal bovine serum was given. Then like the SARS-CoV-2 strand WA-1 was isolated and obtained from the Center for Disease Control, the CDC. The vaccine was then given to baboons and mice. For the baboons, they're given 2 doses, which were given 21 days apart from each other. And the mice received a single dose that varied in size of the SARS-CoV-2 vaccine. So figure 8 shows a humoral and a cellular immune response to the NVX-CoV2373, and it's shown with and without matrix and adjuvant in baboons. So like this first part of the figure just shows the immunization period of first the serum collection, which was at day 0, day 21, day 28, day 35, and then the PBMC collection was at day 0 of the study, and then day 28. The figure shows like the receptor block antibodies in the baboons were like measured through a program called ELSA, and it was shown that there's an increase by 8 folds in the receptor inhibition. So like if you look at here at day 0, it's shown only measured at 8. Then like at day 21, it is measured to have up to 64, which is a times 8 increase. Then if you go from day 21 to day 28, there's another times 8 increase from 64 to 512. And lastly, you look at figure 9, it compares you see a comparison of COVID and of a COVID human convalescent serum antibody levels to the levels in the NVX-CoV-2373 vaccine that was given to the baboons. As you can see, the vaccine that was given to the baboons is comparable to that was given to by the humans. As you can see, there's like little to no error to what was shown versus what they expected. So signs show that it's comparable. So the takeaways from the COVID and all article, so combined with the data that's presented in this study plus other studies that were done on non-human primates and in a clinical trial testing phase, the author states that the author supports the mRNA 1273 vaccine, saying that it passed in the clinical efficacy trials. Colbert goes on to explain that prior research on the candidate MRS-CoV vaccine, it paved the way for a rapid response in a current COVID outbreak. Colbert also writes that this is a demonstration of how the power of new technology-driven concepts like synthetic vaccinology facilitates a vaccine development program that can be initiated with the pathogen sequence alone. So the takeaways from the TN article, based on the findings of TN, it can be said that the vaccine tested on the baboons and mice could work on humans. As it was shown in figure 9 in the article, the vaccine has some comparable results in both humans and baboons. And that is my presentation. Thank you for your time.